So my name is Kevin Caulfield. I'm a PhD student at the Medical University of South Carolina with Dr. Mark George. And today I'll be talking about a collaboration that we've had with Dr. Adam Woods and his postdoc, Dr. Indal Astari at the University of Florida. And this was a study that uses tDCS for working memory. So to just give a little bit of background on the topic, prefrontal tDCS for working memory is an appealing stimulation method, but the results to date are mixed. So there have been 76 studies so far, and 51 of them, about 2/3, have found positive effects, but 1/3 have found little to no effect from the tDCS on working memory. And this might be due to a near infinite parameter space that can differ between studies. So just to list a few that can differ: working memory task, lecture montage, state dependency, patient population, polarity of current, time of assessment, number of sessions, and there are at least 10 others. But nevertheless, even though there are all these differences between studies, there's one common factor in each study, which is that a uniform dosage of 0.5, 1, 1.5, or 2 milliamps is applied for each participant or each patient, and that's irrespective of their individual neuroanatomy. So this is where the electric field modeling comes in. And what we hypothesize is that the mixed effects of these studies to date could be due to variable amounts of stimulation reaching the cortex with that uniform dosing mechanism. And we hypothesized that we could do something that we developed called reverse calculation tDCS modeling, using the electric field modeling to ensure that each person receives the same electric field at the cortex. So this was a 2-experiment design. And in experiment 1, we asked the question, can reverse calculation modeling reduce the electric field variance at the cortex? And in experiment 2, we asked the question, what electric field threshold should we reverse calculate at? And the data that we used were from this NIBS et al. Paper from Dr. Woods' lab in 2019, and it's published in Frontiers in Aging Neuroscience. And it was a cohort of 28 older healthy adults who all had anatomical MRI scans. So first of all, what is reverse calculation modeling? I like this image from this Evans et al. Paper of 2020 in brain stimulation. And to date, there have been 3 published studies using reverse calculation modeling, and what I'll be presenting now is under review. But on the left here for this fixed dose, this is what most tDCS or all tDCS studies are like to date, where there's this uniform input of, say, 2 milliamps, and this produces a variable output electric field at the cortex. What reverse calculation modeling does is the opposite. So it tries to threshold for the output in the electric field in volts per meter and individualize the dose at the scalp to produce that uniform electric field at the cortex. So how do you do reverse calculation modeling? It's a 3-step process. And so step 1 is to run an electric field model. And so here we did it with 2 milliamps of stimulation over F4 for the anode and F3 for the cathode, which is what was used in that NIBS et al. Paper. And so after you get the scan, you segment it and MNI transform it, you create that volumetric mesh, and then you can place the electrodes and run ROI analyses over different MNI coordinates. So here we looked at the left dorsolateral prefrontal cortex. Step 2 is you can create these proportions of the input, so the 2 milliamp dose over the electric field that it produces for each person. And then you can calculate the individualized dose needed to produce any electric field that you want. Here we did the group average electric field, so you can see how we cross-multiplied to get that individualized dose. And then step 3 is that you can repeat step 1 with the individualized dose and look at the same ROI to see if it made a difference at all. So first I'll show you what we see visually. So these are the 28 brains of the 28 participants in this working memory study. And so what I want to convey here is that when you apply that uniform dosage, as you might expect for people with different skin, skull, CSF, white matter and gray matter compositions, that you get some variability between individuals. So some people have higher electric fields from 2 milliamps, some have lower electric fields from 2 milliamps. So what happens when you individualize the dose? What I want to show here is that you get a little bit more consistency. So there's still some variability between individuals, but you can actually calculate it such that each person has that group average electric field at the ROI. So these are paired. So in the blue box, red box, and yellow box, those are the same brains. And if we look at those 3 specifically, those were the people with the lowest initial electric field, the median electric field, and the highest electric field from the 2 milliamps of stimulation. And if we isolate these and look at how they change with the individualized dose, they're all much more consistent after the fact. So the person with the lowest electric field to begin with, when you individualize the dose, the electric field goes up. For the person with the median electric field to begin with, it stays about the same when you individualize the dose. And for the person with the highest electric field to begin with, when you reverse calculate to the group average for the individualized dose, it goes down. What does this look like more quantitatively? So here on the y-axis is the electric field in volts per meter. And in the blue dots, it's the 2 milliamp electric field. So you see this dispersion of electric field from the 2 milliamps. But when you reverse calculate the dose, you can get the exact same group mean. So that's that black bar of 0.221 volts per meter. But the standard deviation drops about 100-fold, so 2 orders of magnitude lower. So I said this was a 2-experiment design, and here's an interim summary. So in experiment 1, we asked, can reverse calculation individualized dosing reduce the electric field variance? And it looks like it can. So now in experiment 2, I'll talk about what electric field threshold should we reverse calculate at, and is there an electric field response curve in working memory? So for the methods, it's the same 28 participants from experiment 1. And here they performed a working memory M-back task. So it's a 2-back task with letters, which is a commonly used working memory task. And the dependent variable was accuracy as a percentage. And this was measured pre-tDCS and post 10 sessions of either active tDCS or sham tDCS. There were 14 participants in each group, but we excluded for ceiling effects. So if someone had 100% accuracy at baseline, we excluded them, and that left us with 12 active stimulation participants and 11 sham stimulation participants. This was a triple blind design. And the key manipulation here to look at our question of whether the electric field matters is we split the tDCS participants in half. So we call this high electric field from 2 milliamps, and those were the 6 participants with higher electric fields than median from the 2 milliamps of stimulation, and low electric field condition for the 6 participants with lower than median electric fields from the 2 milliamps of stimulation. And we hypothesized that the participants with the higher electric fields from that uniform 2 milliamp dose would be driving the working memory improvement from tDCS. So first, I'll just show what does active versus sham look like. So there's a main effect of time and an interaction between time and condition, as you might expect. And with the post hoc analysis, we could see that this effect is really from the 2 milliamp active stimulation condition, such that those participants improve in working memory performance about 14% from pre to post tDCS, whereas sham stimulation participants only improve about 1%. But what gets really interesting is when you break it down into that high electric field condition in red versus the low electric field condition in blue versus sham in gray. So here again, there's a mix, with the mixed ANOVA, there's a main effect of time and a significant time by condition interaction. But the post hoc analyses reveal that there's no differences at baseline, but from pre to post stimulation, the high electric field condition participants seem to be driving that working memory improvement. So they improve about 19%. For the low electric field condition participants, they still see an improvement in working memory of about 7%. And then for sham, again, there's only about a 1% improvement. So to summarize, in experiment 1, I talked about individualizing tDCS dose using SimNIBS reverse calculation modeling to reduce the electric field variance at the cortex. In experiment 2, there appears to be an electric field response curve in tDCS for working memory, such that high electric field individuals from the 2 milliamps of stimulation appear to be driving the behavioral effect of working memory improvements from tDCS. So as a future direction, how do we put these results together? Well, what you could do is prospectively apply these reverse calculation individualized tDCS dosage, and you can do this for each person and maybe choose an electric field in the high electric field range. So for example, in this study, it would be in the 0.25 volts per meter range or so. And perhaps this can improve the effect sizes and the consistency of results from tDCS for working memory. So with that, I'd like to thank Dr. Woods and Dr. Ndal-Story. The funding for this study was provided by Dr. Woods' KO1 and R01 awards. And I would also like to thank my mentor, Dr. George, as well as the Brain Stimulation Lab. And I'm happy to take questions, or feel free to email me at that email address. Thanks.